Hi, good morning, good afternoon, or good evening. So today I am going to be discussing about my critique on the global diversity of Aloricate oligotrichea in marine and brackish seawater, a journal entry written by Sabin Agatha that was published in year 2011 and was funded by the Austrian Science Foundation, who claims to have no participation in the gathering of data. So what is an oligotrich? An oligotrich is a spherical to pear-shaped protozoan of ciliate order Oligotrichida found in fresh, salt, and brackish water. One special characteristic that they possess is that their body cilia are formed into bristles. One thing also that they have is that they have conspicuous adural ciliature, which means that on the groove leading to the mouth is where their cilia is located. Another thing is that they have anterior spiraling of membranels, which means that their cilia lies on a flat plate. So this cilia serves as an efficient way of locomotion, and the organism on the picture that you see is called Strobidium inclinatum. To summarize Sabin Agatha's research paper, her study focuses on the worldwide distribution of ciliated protists in the subclasses of Oligotrichia, which are considered major components of marine plankton. So, oligotrichids and choriotrichids contribute to the multi-step microbial food web and dominate the marine microzooplankton. 141 species and their synonyms were examined and analyzed using ecological and taxonomical data available at that time. So the researchers found out that there are 94 morphospecies that show a worldwide distribution. They have also said that these 94 morphospecies match the modern endemicity model. That means that they are prevalent in one location or area for a long time, but this occurs occasionally. So on the other hand, there are 47 morphospecies that are found to settle on one geographic region, which are found in the Antarctic, the Pacific, and the Black Sea. So although the patterns were recognized, the researchers considered an undersampling might have affected the results. One more thing that they have found out is that the species richness is distinctively lower in the southern hemisphere than in the northern hemisphere. But a disclaimer that the author put was that this might have been a result of undersampling. So for the actual critique, number one, for the writing style and the structure, the writing style of the research paper that Sab and Agatha wrote is very reader friendly. It makes one easy to understand what was the message that they were trying to convey. For the structure, it was also very well thought of, and that each section were labeled properly. For the introduction, well, it was very well written and it gives a background of Aloricate oligotrichia, and they even have sections about the history and discovery, the ecology and environment, classification and phylogeny, and including the morphology of Aloricate oligotrichia. So for the materials and methods, the materials used in this study were only taxonomic and ecological studies that were available at that time. So how did they classify the records in order to be approved and to be included in their studies was according to quality. So number one, it has to be a reliable record from the type or neotype locality accompanied by the original description or redescription. Number two, it has to be more or less reliable records that are supported by descriptions, illustrations, and measurement. And lastly, unsubstantiated records based on uncertain identifications were also included. A similar study conducted by Shan Gao and was published 2 years prior to the published year of Agatha's paper entitled An Updated Phylogeny of Oligotrich and Choriotrich ciliates with representative taxa collected from the Chinese coastal waters used the same methodology as Agatha did. But one thing that made the research um, but made sure that the data or result from the research was accurate is that they gathered samples from from Qingdao, Shandong, northern China, and then they were identified first by literature. Then after that, the DNA was extracted using red extract and tissue PCR kit. The result of that DNA extraction was then um, put side by side and compared to what was compiled in the GenBank database, and then afterwards they are placed or they are subjected to a Bayesian inference, a statistical tool which really showed what organism was dominant or was dominant in number in that area. So if Agatha used the same methodology as Shang Dao and his team did, she would have probably addressed one of the problems of her research, which was the accuracy of her data. Biogeographic distribution was also stated in the paper, and it said, a disclaimer said, that the molecular biography of Aloricate oligotrichea is still in its infancy, hence the genetic diversity presented might have been a result of insufficient taxonomic resolution. So based on the methodology applied, the results of the study were greatly affected because they have only focused on the characteristics of organisms and base how they identify organisms on the characteristics. For me, I do not agree with this method because some organisms do possess same characteristics, but when placed on a genetic level, they are not the same. So right now, new studies about Aloricate oligotrich has emerged. One example would be the one study that was entitled The Phylogeny, Classification, and Diversity of Choriotrichia and Oligotrichia. So in that study, they use a different method. They have two approaches into how they classify organisms. So number one is morphologically. So they identified it through sequences like phylogenetic and morphological support. So number two, they use unidentified environmental sequencing. So number one is single cell sequencing and phylogenetic inferences. So if just like the study conducted by Shango, this methodology was applied by Agatha, then she can ensure that the results of her study were accurate. So in conclusion, the statement of the problem presented by Agatha was great, and the study that she had would be very useful. But then again, um, although the style and the structure are superlative, the methodology needs to be improved. I think that she should combine on I think that the future researchers who wanted to conduct the same study as Agatha did should use a combined or a combination of previous studies or literature that is available and field work. And lastly, in order to ensure the species of an organism that they are trying to identify is that they use DNA sequencing so that the results would be accurate. So that would be all. Thank you.